It's the 14th of December, and you're listening to Kopi Time, a podcast series on markets and economies from DBS Group Research. I'm Taimur Beg, Chief Economist, welcoming you to our 39th episode, which happens to be the final one of 2020. What a year it has been, upended by a once-in-a-century pandemic. Although, as we draw closer toward the end of this tumultuous year, there appears to be some light at the end of the tunnel. And simply because, with record speed, scientists and biomedical companies worldwide have formulated and trialed a large number of vaccines. And a few of them are being rolled out as we speak. So somewhat exciting times. And I think it is fitting that we will devote the last Kopi Time of 2020 discussing that very matter, vaccines. To do that, we're pleased to have with us Associate Professor Jenny Low, a senior consultant with the Department of Infectious Diseases in Singapore General Hospital. She has two other important titles. First, she's the co-director of the Viral Research and Experimental Medicine Center at SingHealth Duke NUS. Second, she is Deputy Clinical and Scientific Director at SingHealth Investigational Medicine Unit. Associate Professor Low has been doing clinical research for almost 20 years, carrying out proof of concept and early phase clinical trials. Her current research focus is on early phase adaptive clinical trials of viral therapeutics and vaccine development, as well as understanding the role of early immune responses in modulating the outcome of infection or vaccination. Dr. Low has been awarded twice the National Clinical Scientist Award back in 2016 and then again in 2019. And these were for her research in host immune response to viral infections. She was a lead author in the first publication that detailed host response to severe COVID-19. Finally, and perhaps most apropos, she is currently leading the lunar COVID-19 vaccine that is co-developed by Duke NUS and Arcturus Therapeutics, as well as leading the clinical trial for the therapeutic monoclonal antibody against SARS-CoV-2 developed by a local biotech company. So yes, we have a very qualified one today, listeners. Dr. Low, welcome to Kopi Time.
Thank you very much, Tamil. Very glad to be here.
Oh, I have so much to ask you, and I hope I can do justice to your deep experience and background with my questions. And I will ask with a very baby-like question. Let's start with talking about vaccines. What are they and how are they developed?
Sure. So in general, vaccines contain the germs or part of the germs that cause the particular disease that we are trying to vaccinate against. To give an example, the measles vaccine will contain the whole measles virus, and the chickenpox vaccine similarly contains the chickenpox virus that we want to protect the person against. And influenza vaccine will contain usually will contain the killed influenza virus. So these germs, they can be either killed or they can be weakened to the point that they don't make you sick when you are vaccinated against them. Some vaccines will only contain a part of the germ, so they will not contain the entire germ, but they may say contain just a specific protein of the germ. And our immune system in general consists of two arms, a B cell arm that is responsible for producing antibodies. And we have a T cell arm which produces T cells which will help our antibodies fight the disease. And a vaccine works by stimulating our immune system to produce immunity such as antibodies, and for some vaccines will also elicit T cells, and so that the immune response to the vaccine is the way we want it, as if you're actually exposed to the real disease. And after you get vaccinated, you will develop the immunity or protection against that disease without having to first get the disease. So therefore, very simply put, what the vaccines do is they will reduce the risk of us getting sick from the germs that cause these diseases by training our immune system to recognize and to fight the pathogens such as, it can be viruses, it can be bacteria.
Right. And of course, today we have far deeper understanding at the cellular level of, or even the protein DNA level, how these things work. But of course, long before we knew that, we were trying out vaccines a couple of centuries ago. But since then, the basic understanding has changed, but the basic sort of idea that, you put in some weak part of the germ to develop immunity has still been more or less the same. Now, if we have been developing vaccines for centuries, why does it still take so long to develop? I'm not talking about the current vaccine, but at least the ones that we have seen in the last half century. We talk about years, if not decades, for those to develop. And if those vaccines have taken so long, how are we managing to roll them out so fast in 2020?
Right. Yeah, very good question. So I would tackle the first part of the question first. Why does it take so long, years to develop a vaccine usually? And that's because of the conventional way that vaccine development has always undergone in the past many decades. There are multiple steps involved in the development process, and clearly, this is needed because we want to make sure that when we introduce something new, like a new vaccine or a new drug, that it is introduced in a safe manner and that it's been proven to work, and then it can be introduced to the general population. And this process that I'm talking about, we can very broadly classify them into 3 to 4 phases. So there is the discovery phase, where scientists will first need to discover the vaccine or a drug. Then there is a preclinical phase where the vaccine will have to be tested in animals, first in cells system, then after that in animals. And usually, it needs to be tested in more than one species. And the whole purpose of doing the cell system, the animal testing, again, is to make sure that we address the issue of safety and efficacy, meaning whether it actually works, before we can then move into the clinical phase. And the clinical phase is then broadly divided into clinical phase 1, 2, 3, where the vaccine is then tested in humans, but in a very gradual manner, starting from phase 1, where it is only given to a very small group of generally healthy volunteers, adult healthy volunteers. And at this stage, the aim is to test for safety, to make sure that it is safe in this small group of healthy adults, before it then moves into phase 2, where the vaccine is then now administered to a larger group of people, usually in the hundreds. And these are people who usually have characteristics similar to the ones for whom the vaccine is actually intended for. And again, even in phase 2, when it is tested in hundreds of people, the focus is still primarily on safety, but there is now an increasing focus on the testing for immune response and measuring immune response to the new vaccine. And finally, when it has passed through phase 2, only then can the program roll into a large phase 3 trial, where it is now, it will now take place in multiple countries, in multiple different people of different backgrounds, different genetic backgrounds, different characteristics. And in phase 3, then the focus will then be looking at the efficacy of the vaccine to see if the vaccine will actually protect these people against the disease that we are studying. And of course, at the same time, data will be collected to continue to evaluate for safety. And for most vaccines and drugs, after the vaccine has passed all these clinical trials and is now being licensed and marketed, there is something called phase 4 post-marketing surveillance. And again, because under trial condition, there are very strict criteria. And these conditions may be very different in phase 4, where it is now marketed, is now ready, easily or readily available to anyone, that there may be rare side effects, or what we call rare adverse events, that never came to light in the clinical under clinical trial condition. So phase 4 monitoring will then happen to monitor for such events, and also there's another purpose of phase 4 is to then now allow us to look at the very, very long-term effects of the vaccine in large general population. And usually what happens is all these steps from discovery to preclinical to clinical, they all will happen in a sequential manner in peacetime. So under normal circumstances, each developmental step is performed in sequence. But now, so come to your second question, how is it such that we can now do it so quickly? But that's because in an outbreak, like a pandemic situation, in public health emergencies, there is an allowance for such process to be done in an atypical or in an expedited manner. So I think it's very well illustrated in the way public health systems have responded to the COVID-19 pandemic, where you see that governments, government agencies, international counterparts, non-profit organizations, pharmaceutical company, academia, all come together and to develop a coordinated strategy to prioritize and to speed up the entire development process for the most promising vaccines or even treatment, therapeutic drugs. And what we also see that is happening right now, actually since the beginning of the pandemic, is many governments will then make investments in the necessary areas, for example, manufacturing capacity. And these investments are made at own risk. These then give the companies or the pharmaceuticals the confidence that they can now invest aggressively in the development of the vaccine or the drug. And hopefully that, once it's proven to be safe and to work to prevent COVID-19, then it will allow for faster distribution when that vaccine is eventually approved by the health regulators. And during a public health emergency, there are already existing criteria that have been laid down by agencies that if once certain criteria are met, then the manufacturer can actually submit something called emergency use authorization. So we have heard of this EUA very often now in the media. And EUA then allows the regulator, for example, FDA, to then facilitate and to review the document and to see whether the vaccine can be made available earlier ahead of full approval timeline, which usually would take longer. Yes, so EUA has come in very, very useful, at least now, for the trying to get earlier approval for some of the vaccines, two of the most, the forerunners of the COVID-19 vaccines to get earlier approval. So strictly, if you think about it, then there's actually no predetermined timeline for vaccine development, right? It's just that, typically, if there's no pandemic, there's no urgency, then we will want to follow a more conventional, regulated pathway. And in emergency, then there are contingencies for such processes to be sped up. And even for the clinical trials, increasingly, we are not we are in the need to have rapid response. We are also collapsing some of these phase 1, 2, 3 sequences, and there are something called adaptive trial designs for clinical trial designs, which will also allow clinical trial decisions to be made much faster in a much shorter time, involving fewer subjects or fewer patients. And the whole goal, really is just to shrink that timeline. But at the same time, the statistical rigor is not compromised. Yeah, so I think so. That's my take on how we can do it so quickly in 2020. From the time we knew about the outbreak coming out from Wuhan, China, in early 2020 to now we're just at the beginning of December. There are already 2 vaccines that look like Pfizer have already gotten FDA approval, and UK has started vaccination, and they are also rapidly evaluating the Moderna vaccine for emergency use. Yeah, so and there's a lot of coordination and massive efforts by multiple stakeholders.
Absolutely, like a proverbial Manhattan Project, and I guess that's what humanity needed this year. And Dr. Loh, it is critical that what you have just said is absorbed by the general public, because I'm sure you have been exposed to all sorts of social media discussions, especially in the West, about people questioning the veracity of the process and worrying about the risks. And I think that the key takeaway from what you just said to me is that there has been no sacrifice of statistical rigor or any methodological compromise. And I think this is what I think not only is your responsibility, but also my responsibility to get it out to the public, so that as we roll out vaccine, that people not have any misgiving whatsoever. Now, you just alluded to 2 vaccines that have already been sort of, gone through the trials and are now in the process of being approved around the world. My understanding is also that, some of these vaccines are not your typical run-of-the-mill vaccines, and we have some new exciting technologies in vaccine development. So would you walk us through that?
Yeah, so I think to understand the new development, it would be good for me to maybe talk about the different technologies available in vaccine development. It's been around, either very, very old technologies or newer ones, and of course now the very new mRNA vaccines, right? So it'd be good to have to give a broad overview of the different technologies. And even for COVID-19 vaccine development, actually there are many different platforms or technologies that are being used by different pharmaceutical companies. And so the very well-known the ones that have been around for a long, long time are the live attenuated virus vaccine. So these are your, like the measles, mumps, rubella vaccine that all of us are injected with when we were little babies, chickenpox vaccine, the yellow fever vaccine that is mandated if we visit certain parts of South America or Africa. So these are all live attenuated, meaning these are live viruses, but they've been weakened in the laboratory so that when we are injected with these live attenuated vaccine, they will not make us sick with the original disease, but they will give us a very mild form of the disease so that we will develop the immunity against the disease of interest, whether it's yellow fever, measles, or chickenpox. Now, then there's the killed whole virus, and the one that we are very familiar with will be the influenza vaccine that we receive every year. And hepatitis A, rabies vaccines also belong to the same, also made using the same platform. So these viruses have been killed, so they cannot multiply in our system after we receive this sort of vaccine. But what it does is, the body will still recognize that the virus has been introduced in the system, and it will mount an immune response against the vaccine. Of course, we all know that the flu vaccine, we need to get the vaccine every year to be, because the previous one may not work if there's a change, and for an inactivated vaccine, generally it's not so long lasting. And then there are other platforms, like sometimes we just use a part of the protein or a part of the sugar from the germ. And so like the pneumococcal vaccine can be made from such platform, or the hepatitis B vaccine is a subunit vaccine. And but essentially what all these vaccines would do is, they are trying to train the immune system to recognize part, either the whole of the pathogen or part of the pathogen, and produce the necessary immunity, like antibodies, so that when we are really exposed to the virus or the bacteria, we have these. We will be able to mount the necessary immune response before the germs can make us sick. Now, the newer ones will be, before I go into mRNA vaccine, there's actually quite a new platform, and that's a viral vector platform, and that's the one that AstraZeneca, in partnership with Oxford, have developed the COVID-19 vaccine, and that's using a viral vector. So it's really quite clever. They take a different virus, which usually will not make a person sick. Then, so this other virus, what we call the safe virus, will then serve as the delivery platform to deliver the protein of the virus that we are interested in, or the germ that we're interested in for the protection. So the protein is then built into this safe virus and delivered to the person as an injection. And in that way, and that's how our body develop the immune response to this protein that's been slotted into the safe virus. And so AstraZeneca vaccine, for example, is using this platform. Then, we've read Moderna, Pfizer vaccine is mRNA-based vaccine. And this is really very new. No mRNA vaccine until now have gone into human use in the general population. So for mRNA, this is a nucleic acid, and so it's a genetic material. And essentially what it does is that the genetic material codes for the protein of interest. So in this case, for the COVID-19 vaccine using mRNA platform, the protein is the spike protein of the virus that is introduced into the vaccine formulation. This is what is in the vaccine is actually the genetic material that codes for the spike protein. And once it's injected, it will then, from genetic material, it will then be copied to make protein. Then once the protein is made, then our body will then recognize the protein as a foreign material, and we will then, the immune system will then trigger the body to produce antibody against the protein, and that's how the mRNA-based vaccine technology induce immunity in people who receive the vaccine. So I think what we can see from the different technologies is every platform will have their pros and the cons. Live attenuated, for example, generally will give you very, very long-lasting immunity, sometimes like lifelong. Just one injection of yellow fever vaccine, you will have protection against yellow fever for is lifelong durability. Then some of the protein-based platform may need multiple revaccinations, and certainly with the inactivated, I said earlier, some of the inactivated vaccine will not be able to induce long-lasting immunity, and therefore may need repeated vaccinations. Now, for mRNA vaccine, there are a few advantages that come to mind immediately. Is that it can be readily ramped up. It can be produced in large quantities very quickly, and certainly, for pandemic response, that's really ideal. Because, for example, some of the live attenuated vaccine or inactivated vaccine may be made in eggs. Would require the vaccine to be developed in the eggs, and that can be really slow, and you won't be able to get large quantity very quickly. Yeah, so mRNA vaccine have the advantage of really rapid development. And the other good thing about this nucleic acid platform is that we now know that it can induce very strong T cell immunity. So not just antibodies. Early on, I said that we require our immune system has two arms: the B cell arms, which will produce the antibody, and then the T cell arms, where the T cells will actually help the antibodies, will help make very good antibodies. So nucleic acid platform we now know can induce very, very strong T cell immunity.
That is good to know. And I think you have already anticipated a question that I wanted to ask later on. But just to stay with the mRNA vaccine, so we've got the Moderna project and the Pfizer project. Are they almost identical, or they're a bit different from one another?
Yeah, so Moderna and Pfizer vaccines are fairly identical. They are mRNA-based vaccine platform. But the AstraZeneca's one is a viral vector vaccine. So the virus, the safe virus that I spoke about just now, in the AstraZeneca vaccine, this safe virus is actually a chimpanzee adenovirus. And they then deliver the SARS-CoV-2 virus protein, the spike protein, in the chimpanzee adenovirus. And then it's delivered as a vaccine injection to people.
And how does one extract that from a chimpanzee?
Taimo, can you repeat your question?
And how does one extract that safe vaccine from a chimpanzee?
So it's cultured. So it's grown in the laboratory. Okay.
Because I had read somewhere that it comes from the excrement of chimpanzees, so I wanted to, clarify that with you.
I believe because once you know the sequence of the chimpanzee adenovirus, technically you can just grow the virus in the lab. So usually these viruses will be grown in the lab, in the laboratory under very strict requirement for vaccine production.
Right. So the visual that one would have to go to the zoos to find all the chimpanzee is not quite in line with reality. I'm really glad and relieved to hear that. Dr. Loh, so beyond the Western vaccines, of course, we've been reading about many vaccines under development. The Russians have the Sputnik 5, and of course, perhaps a bit under the radar in the Western media is that there are several vaccines under development in China, which have also been trialed and being administered fairly widely. So what's your sense of where those vaccines stand?
Yes, so China has several vaccines under development, and at least I think 3 of them must have gone into phase 3 clinical trial by now. We do know that most of the China-made vaccines are developed using the inactivated whole virus technology. So very similar to how, say, the influenza vaccine is made. It's the same kind of platform. What I know is what I read from the media, and we know that they have been very aggressively vaccinating their population, and more than 1 million people have been vaccinated in their trial by now. They are also conducting the trials in countries outside of China, so Indonesia, Brazil are just a couple of them among the, I think, more than 10 countries that are participating in the Chinese clinical trials. But I have not seen any scientific data yet. I believe at some point those data will be released in the public domain. But I'm quite sure by till now those data have not been published yet.
Okay, because I remember a couple of weeks ago, the Indonesian government was announcing rather enthusiastically that one of the Chinese vaccine trials there have had a high effectiveness rate. And then the Chinese authorities themselves said that, no, we don't have, detailed enough data to, go for such high effectiveness numbers. So yes, we are all sort of staying back from declaring victory on those things. Dr. Lowe, these vaccine effectiveness numbers, so in the last 1 month, all of us have been exposed to all these 90%, 95% effectiveness numbers. What do they mean and how are they calculated?
Yeah, it's really interesting to read about such high efficacy numbers, and I think certainly it looks really, really promising from what we are reading in the press. Now, vaccine efficacy is simply the percentage reduction in the disease in the vaccinated group compared to the unvaccinated group under optimal conditions. So in this case, we are referring to these optimal conditions we are talking about is the phase 3 clinical trial condition. Now, so whenever we talk about efficacy, this is the result obtained under trial condition. Vaccine effectiveness is what is the ability of the vaccine to prevent the disease in the real world when it's after it's been licensed, marketed, and is given to the general population, not under a trial condition, so in the real world. So essentially, they measure the same thing. They measure the rates of disease between vaccinated and unvaccinated. And it's just the efficacy is the word we use when is measured under controlled clinical trials, but effectiveness is measured when it is already approved for use in the general population. And from these numbers or this percentage, we can then identify, or we can at least postulate what is the proportion of vaccinated people that we would expect to be protected by the vaccine.
Right, that's a critical one. So, Dr. Lowe, I understand that, these large-scale phase 3 trials, you'll have 20,000 people in placebo, 20,000 people in control, and then the experiment would end at a certain end number, that if 100 people have developed symptoms of COVID-19, then we will now see, how many of them were placebo and how many of them were in control. And how does one come up with these end numbers? Is it just based on long-term experience, or there's some science behind that?
There is a lot of science behind that, and that's taken from, so that to even come out with those estimates, usually we will need a statistician to work with us, and using existing evidence in terms of the prevalence of the disease in the particular place, how many cases we would expect to come down, how many people will expect to come down with the disease over a particular time frame, say 3 months, 6 months, or 1 year, and based on our understanding of the vaccine, how much protection we think we will expect to see from someone who have received the vaccine, then the number is then calculated. There are formulae that we work with to then calculate what is the size of the study population that we will need to enroll in order to see the expected number of people who will get the disease and versus the number of people who will not get the disease in the so unvaccinated versus vaccinated, what proportion we will expect to see. So that requires certain statistical calculations back end before the trial even starts. Sure.
Why are some vaccines single dose, and at least the ones that we have seen out of the West now seem to be mostly 2-dose vaccines? And then we heard some stuff about the AstraZeneca one. The best one is a 1.5-dose vaccine.
I think I wouldn't comment too much about the AstraZeneca vaccine dose, because that was a finding they made after they realized that there was an error in the dosing. regimen, and that wasn't even the intended dose that they had wanted to give their participant at the beginning of the trial. But in general, a lot of the vaccines we use these days, they are quite new, and there's always we need time and we need follow up to understand what is the length of time that these vaccines can provide the protection that we're hoping the vaccine will be able to do. So, for example, a lot of just by natural infection, right? For many diseases, immunity will develop after the natural infection, but it will also wane. And same thing with vaccines. The durability or the duration of the immunity that's elicited by the vaccines will vary. And what causes variation? It depends. It depends on the type of vaccines. It depends on the host. So the person that received the vaccine, right? Whether it's a young, very, very young person, a very old person, the duration of the immunity, again, may make a difference. And how the vaccine is given, is it given, if the interval also matters. Sometimes, if the interval is too short, then the durability of the immunity may also be affected. And for a lot of vaccines that require more than one dose, there's a minimum interval that is needed in order to achieve the best outcome. Then earlier on, I spoke about the different platforms, pros and cons. So live attenuated, we know, can usually give very long-term immunity, much better than the subunit vaccines, for example. So subunit vaccines very often will require frequent booster doses, even after the initial primary series. And for vaccines that are made of, that use the sugar of the virus, usually such vaccines will not produce long-lived memory cells. So this kind of vaccines also usually will not give long-lasting immunity and will require repeat boosting.
Will we have to take vaccine every year, like a flu shot?
So we don't have an answer yet, and a lot will be dependent on how long the immunity will last. So these will then require us to follow up the trial participants in the long term, especially those from the phase 3 trials. And also from post-vaccine licensure, we will have to monitor people who receive the COVID-19 vaccine in order to determine whether they will need repeat, whether there will be waning immunity against the vaccine, and or how long it would take before the immunity starts to wane, before we will know if they will need a repeat dose, or even how often the repeat dose should be administered.
Right. And I think that's the big question around that right now, when winter comes, especially in the colder parts of the world, we take a vaccine, we get immunized for flu shot, and that also doesn't really last more than 6-8 months. And to expect much longer, as you said earlier, it depends on the construction of the vaccine and how the long-term effects are. Now, I want you to sort of dispel a couple of urban tales that are out there. As you know, social media, lots of theories out there. One is that some of these vaccines, the way they're being designed, could lead to patients becoming an asymptomatic carrier. Is there any veracity to that?
So there isn't. No, this will not happen. Let's take the mRNA vaccines, for example, whether it is Moderna, Pfizer, or the Arcturus vaccine. What is introduced in the vaccine is the genetic code that codes for the spike protein. There will be no virus. the vaccinee will not be able to make a whole virus after the vaccination, because only a protein, only a part of the germ, the protein, genetic material, is introduced into our body. So there's no risk that this protein that's made will then grow, will then assemble into an entire virus and become live and start multiplying. That biologically, scientifically cannot happen. For the Chinese vaccines, those are killed in activated vaccine. So those viruses, even after it's been injected into our body, will also not be able to multiply and make more viruses. For the AstraZeneca vaccine, again, although we use there's a safe virus, the Chimpanzee adenovirus, that is acting as a delivery system to carry the SARS-CoV-2 viral protein into the body. Again, it is the protein that's being delivered into the body. They cannot assemble into the entire SARS-CoV-2 virus. That's biologically it cannot happen.
But let's say I have been immunized by the mRNA vaccine, and then I'm exposed to a huge amount of SARS-CoV-2 viral load. Would the vaccine elicit the type of antibody response that destroys those incoming germs, and therefore I will not be a carrier?
Okay, now I understand where your question is coming from. No, that's a very good question, and certainly a very valid one, because what we do know now is the vaccine can protect against severe disease. So can the vaccine prevent infection is the question.
That's right.
We think that it is possible. It is likely that even in someone who is vaccinated and has immunity against SARS-CoV-2, if this person comes into contact with the virus, it is highly likely that this person can carry the virus but does not fall sick and will not develop COVID-19 disease and will not get severe disease. So yes, it is highly plausible that they can carry the virus, but they do not develop the disease, and there is a possibility that they can still pass the virus on to someone else who has not been vaccinated and therefore can potentially still fall sick from being exposed to the virus. So that's why I think it is important that, other than vaccinating as many people as possible, that we need to continue to have treatment options for people who, for whatever reason, cannot get vaccinated or who don't want to get vaccinated, and they may still get infected and can still come down with the disease. To get a vaccine, I personally think to develop a vaccine that can totally stop infection. That means the moment the virus gets into our system, if we are vaccinated, the virus just have no more ability to multiply. I think that is a very high bar to reach currently. At some point, if we can fully understand the mechanism, and what it is that it takes for the body for the virus to stop multiplying in our body, we may be able to develop a vaccine that can totally even stop infection, not just disease, to even stop infection. But that's tough, I think, for vaccine development. So we will. Yes.
Sorry, please go ahead.
Yeah, so that's why I think the approach vaccine is not going to solve the COVID-19 problem overnight, and it's not going to the virus is not going to disappear because of vaccine. And that's why it is important that while vaccine will help to certainly control the pandemic and reduce the reduce death, reduce the number of people developing severe illness. The fact that we cannot totally eradicate the virus through vaccination would mean that we need to continue to make sure that we develop effective drugs to treat the disease for people who will still come down with the disease and who may still get very sick from the disease. They will need treatment options as well.
Dr. Lo, that is such a critical part. And I really thank you for that very clear exposition, because that has two major implications from a public health perspective. One is that we have to widen the vaccination net as much as possible. But secondly, that just because we have vaccines rolling out doesn't mean that we can just resume normal lives. We should still maintain all the precautionary protocols that we have taken this year into next year in terms of wearing masks. That even if I have been vaccinated, that doesn't make me completely, I may be immune, but I still have a social responsibility and I should still wear a mask. I should still maintain social distance because I could potentially be an asymptomatic carrier. So I think, again, it's so important for that sort of messages to go out as we start rolling out the vaccines, because my fear is that some people will say, I am immune and I'm like a Superman and I don't have to maintain any social distancing and I don't have to wear any mask. And these sort of messages are very, very important for the community. So I thank you for that. Related to this question is the notion that a vaccine should give you longer lasting immunity than getting the disease outright. Is there an accurate difference between the two?
No, I don't think we. So I tackled part of this question earlier on in terms of whether the vaccine is going to offer long-lasting protection or whether multiple doses are going to be needed. Yeah, so we do know that you will develop natural immunity. You will develop immunity after a natural infection, but because the virus has been around in the human population for such a short time. It's only been a year or so that is circulating in the human race. So we really don't know enough at this point how long natural immunity will last. If we take SARS outbreak in 2003, we are about 17 years post SARS outbreak. And we have evidence that in those individuals, even after 17 years, we are able to detect T cells that can still recognize the SARS virus. Whether the same is going to be true for SARS-CoV-2, we don't know. Only time will tell. And, even for the SARS-CoV-2, the SARS patients whom we can still detect T cells after 17 years, we are also not sure if these T cells will, the presence of these T cells will be able to protect them against another episode of SARS. We think they will be protected, but we don't know for sure. And I think, the same applies for SARS-CoV-2. Till now, we do know that, because it's been about a year, we have been following up patients who have come down with COVID-19 since the beginning of the year. We know they continue to have memory against SARS-CoV-2. For the time being, they probably will be protected against a second infection. How long it will last, nobody knows. Again, only time will tell. You have read, I think in the news on and off, we read that, there have been second infections, but it's been very, very rare. Such occurrence of second infection from SARS-CoV-2 have been very rarely reported. So, I believe the people who had it once for the time being at least, they have some protection against a second infection. The vaccine, like I said earlier on, we really don't know how long, whether it's the Moderna vaccine or the Pfizer vaccine or the AstraZeneca vaccine. At this point, we really don't know how long the immunity will last. And so for, the pharmaceutical companies will have to continue to follow up the trial participants in the coming years, to repeatedly test their blood for antibody and T cells in order to determine the long-term immunity of these vaccines. Because it's been so new, and the trials have just completed. Many of them have only been on the trials for months, less than a year. So these are all open questions at this point.
Right. So professionals in your field have years, if not decades, of work ahead. Dr. Lowe, the distribution of vaccine is going to become the big issue for 2021. Can you sort of give us a sense of the scale of both the manufacturing and the logistics of distribution that's ahead of us?
Yeah, so maybe I'll just focus more on the trial completion, approval, manufacturing.
Sure.
I think logistics, is a really complicated process as well, and I'm probably not in the best position to talk about logistics issues. Now, so if just approval and manufacturing on their own are really, really complex processes. In phase 3 studies, because the trials are run in multiple sites, multiple countries, involving 20, 30, 40,000 participants. So usually these vaccines also have to be made in what we call lots or batches. And this is also part of the process where the manufacturer is also beginning to ramp up for commercial scale manufacturing. And the regulators are already at this point at phase 3 will require the vaccine manufacturer, to submit data to support this whole manufacturing chain activities, and to demonstrate that they are able to produce different batches to the same standard of consistency. And so there are multiple batteries of tests that they'll need to run, and they will need to provide the data and to prove what we call the lot-to-lot consistency. And then the regulator will examine the data. They will even examine the facility, the operations, and for compliance, etc. And once that can be reliably and consistently shown, then together with all the preclinical, clinical development packages and programs are all compiled, completed, the pharmaceutical company or the manufacturer will then have to submit a license application to the regulator or agency, and that's to seek the necessary approval, licensing approval for the use of the vaccine to be marketed and to be used in the general population. And so when all that's done and the health regulator is then happy, with the evaluation, and then the product, in this case the vaccine, can then be approved. Usually it will take months, but as you can see under EUA condition, it can be done in weeks, which recently we have seen with how FDA have reviewed and approved the Pfizer vaccine. And then once this is done, then there is still the phase 4, which I talk about later, and especially when a product or a vaccine is rolled out so quickly, that all the more the phase 4 post-licensure and post-marketing surveillance for side effects is even more critical and essential. Yeah. So this is what it would take for a pharmaceutical or company or manufacturer to go through just to get approval for large-scale manufacturing of the vaccine. Then, of course, then down the line, as you have alluded to, there are multiple logistics issues that will have to be dealt with in terms of distribution. And we read in the news a lot said about the logistics of distribution because of the cold chain needed for the mRNA vaccines, because they need to be transported in -70, -80 degrees kind of condition, which again, adds complexity to distribution and widespread vaccination. Yeah, so a lot of from what I have been reading is there's a lot of coordination needed to make sure that this cold chain can be maintained, and that all the manufacturing raw material have to be made available for the packaging of the vaccine, then the transport of the vaccine, then finally to the end user to be vaccinated. It's an enormous task. It's mind-boggling to even think and to read about it.
Right. I mean, it's an enormously complicated and enormously costly task. I mean, Dr. Loh, the way I think about it is the cold chain that you're talking about. How on earth would poor countries, set up a chain where they can ship, -70 degrees Celsius, containers across, distant countries? You are, of course, familiar with the COVAX initiative, and you have been seeing, what the World Health Organization is doing. What's your sense? Would the, less prosperous countries of the world also be able to receive vaccine at a reasonable rate? And are there sort of serious initiatives underway for 2021?
Well, I think in the short term, we all recognize that not everybody will get the COVID-19 vaccines, and from the word go, and there has to be certainly prioritization. And again, because it's approved under a EUA sort of condition, then we need to understand that actually there are multiple factors, right? That we have to take into consideration whether we should be administering such a vaccine to a person. Certainly, I've said earlier on, we don't have enough data to prove the long-term sustainability of the immunity that any of the current COVID-19 vaccine will be able to confer. Then, there are safety issues at stake for different segments of the population. A lot of the trials that have been done on healthy volunteers, maybe some elderlies, with comorbidities, but there will be segments of the population with unique health conditions that may not have been trialed in the previous studies, and so there will always be safety issues that healthcare providers will always be concerned about. And like I said, this is a EAU, sorry, and there's no formal approval yet, and so clearly, at the EAU stage, then the vaccines obviously, have to be prioritized really to the people who most need it at this point. And long-term, side effects, again, is unknown at this stage. And so I think it would not be wise to just freely roll it out. And certainly most countries are taking a stepwise approach as well. And in Singapore, as far as I've been made aware, is that the frontline healthcare workers will be priority, and certainly the elderly, the very elderly will also be will be prioritized to receive the vaccine once it's available in Singapore. But Having said all that, right, I think the scientific issues aside, we have to forge ahead with the introduction of the vaccines, as long as, again, it has undergone rigorous clinical trial evaluation, regulated evaluation. We know that the vaccines are deemed safe and has been proven to work in clinical trials, then it should not stop us from using them. We just need to know how to prioritize. Your question about WHO, and the role of WHO, and some of the difficulties we have with these mRNA vaccine requiring very unique cold chain conditions and all that. I think as far as this unique cold chain condition issue has come about, then it really brings up the important point that then all the more we need to have availability of vaccines made under different technologies, so that for example, the AZ vaccine can be stored in normal refrigerator condition, so 2 to 8 degrees, and the vaccine is stable in such a temperature range. Yeah, so it'd be good once the different pharmaceutical companies start to release their data and we have more options, then the world will also have more options, and different vaccines will probably be suited for different countries depending on the infrastructure, the capacity, the needs of the different countries, and so you mentioned COVAX, and so certainly COVAX. That's what COVAX. The whole purpose of this COVAX facility is to make sure that the resources are pooled, so that to support the procurement and equitable access, to the vaccine for especially the lower income countries. And already different countries, I think, have been asked to come out with their vaccination plan, and advise. WHO has a SAGE committee, so this is the strategic advisory group of experts, right? We will advise on immunization, and these countries have been asked to come out with the vaccination plans, and SAGE has been advising them on how to on some of these issues. And WHO has, very broadly already prioritized who should be the first group of people to receive the vaccines. And so, no different from how Singapore is doing it. It's going to be frontline personnel and the seniors, who will be the first in the queue when the vaccine is available in these countries.
All right. And the queue is going to be, I think, the feature of the first half of 2021. Dr. Loh, I was just reading about a small state like Alabama in the United States has 300,000 frontline healthcare workers. And I was listening to an interview of their Surgeon General that their first delivery of the Moderna vaccine is only 40,000. So therefore, they can't even cover their healthcare workers, let alone the elderly, and the rest, of course, is going to be down the road, unless, as you correctly pointed out, there are other vaccines coming into the pipeline, and then one can sort of get different quantities of different vaccines and start prioritizing. No, from a public health management perspective, an exceptionally daunting year is ahead. Now, you are leading the effort to develop the lunar COVID-19 vaccine right here in Singapore. Tell us about how that is going.
So this is the Arcturus therapeutics vaccine. We call it the lunar COVID-19 vaccine. It's co-developed between Arcturus and Duke NUS Medical School, and SingHealth conducts the clinical trial since the clinical trial started in, August this year. I remember it was just before National Day. So we have finished the initial phase 1-2 of the clinical trial, and 106 participants received either the vaccine or a placebo. And we have excellent safety profile from the clinical trial. There was no serious adverse event that we encountered. And 28 received a placebo. The rest of them either received a single dose of the COVID-19 vaccine or a double dose. And of the 78 that received the vaccine, we had positive results in terms of antibody and T-cell response in 77 of the participants. So the trial is now on track to start phase 2 end of the year. And phase 2, we are looking to recruit 600 subjects, but phase 2 will involve Singapore and US. And we hope phase 2 can be finished within 2 to 3 months, which will then allow us to rapidly roll into a large phase 3 trial. And phase 3 trial will definitely be multi-site, multi-country.
Well, I wish you the very best of luck in this, highly critical work. Basically the world's fate depends on the success of professionals like you, Dr. Loh, nothing less than that. Again, thank you for such an informative conversation. I think myself and my listeners, we learned a tremendous amount, and we're also humbled by the daunting nature of the work involved and the days ahead. Dr. Jenny Loh, thank you so much.
Thank you, Tamu.
Thanks to our listeners too. Kopi Time was produced by Martin Taki. Daisy Sharma and Violet Lee provided additional assistance. It is for information only and does not represent any trade recommendations. All 39 episodes of Kopi Time are available on YouTube and on all major podcast platforms, including Apple, Google, and Spotify. As for our research publications, webinars, and live streams, you can find them all by Googling DBS Research Library. We thank you for your time and listenership during this year like no other. We'll be back next year with plenty more insights into markets and economies as we try to put the pandemic behind. Happy New Year.